Hi, I'm Connie Lee. I'm the president and CEO of Angioma Alliance, and you are here tonight for a webinar with Dr. Awad and with two members of his team. This evening, we're going to be talking about the PIC 3CA research, a continuation of what we started last week with Dr. Khan and Dr. Marchuk, including some more information about rapamycin. We're going to be getting updates on the Atorvastatin trial, probably some updates on biomarkers, and Dr. Awad is going to answer questions, and he's probably going to throw some other things in there as well that I didn't mention. I just would like to introduce him first, and then he can get going. Dr. Issam Awad is the University of Chicago's John Harper Seeley Professor of Surgery. He leads the neurovascular surgery program and co-leads the stroke and neurovascular team. He's the director of our CCM Center of Excellence in Chicago, and he's the chair of our scientific advisory board. And I would like to welcome you, Dr. Awad, again.
Thank you for getting us together.
Go ahead.
So let me go ahead and introduce Justine Lee. She is our research nurse, and you guys had worked before with Christina, and Christina has moved on to becoming a manager at our institution, and it's taken two people to replace Christina. So Maria Aid remains the clinical coordinator. So for people who are seeing me for clinical reason, for consults and follow-up and so forth, you will be hearing from Maria. For people who are seeing me for research purposes or a trial, or to ask about the trial, or to follow up in a trial or biomarkers, you will be hearing much more from Justine. And of course, you all know Agnieszka, and Agnieszka can wave. So Agnieszka really has recently been promoted to manager of our clinical research, which has increased about 5 or 6 folds in the last 2 to 3 years, and it's been an amazing feat for Agnieszka to take on all these tasks with multiple clinical projects, and we are very happy to see her smile and her abilities to continue to interact with you and to help us with that. So they will weigh in if there are any questions that are appropriate for them. So Connie asked me to kind of give a little bit more of a translation or a clinical explanation of the PIC3CA discovery. This is a Nature paper that came out that many of you have already known about. And I know that you had a webinar with Dr. Khan and Marchuk, and I must have received 1,000 emails afterwards from people asking, can you explain to me what Dr. Marchuk and Khan said? So I spoke with Dr. Marchuk. Dr. Khan is on vacation, but I spoke with Dr. Marchuk, and I got a bit of an idea of what he reviewed, and I want to explain to you a little bit what we discovered in that paper and what it means clinically. And also everybody at the same time focuses on this issue of, should I be on rapamycin? So I have received many calls, can you prescribe me rapamycin? So the question of, can we use rapamycin for CCM, and if not, why not? And using this also as a way to explain how we take drugs that have effects in the mice and effects in the dish, and what is the process that we take to bring them to patients. And we've talked a bit about it before, but now we have several drugs hanging out there, and I'll try to give you a little summary about how this process is playing out for these different drugs. And finally, I would be amiss not to update you on the great progress we are doing with the Atorvastatin trial, that we are approaching the final stretch in enrolling patients. And We need to continue to follow everybody who's enrolled. So do not get tired and make your appointments and keep taking your medication. So we find an answer. But I have some updates on that trial as well. So first of all, Connie, does this mean like a reasonable agenda for about half the talk, and then the rest will be Q&As?
Absolutely. And I wanted to thank you for letting me have one more word, because I forgot to remind people, if you have questions, to use the Q&A at the bottom of your screen rather than the chat, because we may not see it in the chat. We'll see it in the Q&A.
And I want to also say that, you know, I'm so honored when I see these webinars where we have 71 participants, and you know, so I really value your trust in what I'm gonna try to explain to you. So we've always known that people have cavernous angioma, and some lesions stay fairly quiescent and other lesions take off like crazy. And people know that lesions they can have one big one and many other small ones. And they ask me every time, what makes this one lesion go very big while the other lesions are small? Now, we've known that there are factors that can drive all the lesions, things like pro-inflammatory factors. Some of us are born with pro-inflammatory tendencies. Others have a pro-inflammatory state because of vitamin D deficiency. And those people, in fact, have more lesions and bigger lesions and more bleeds. We've known that for some time. And we've also known that the microbiome, which everybody who sees me knows that we talk a lot about it during the clinical visit, because it's a very powerful factor in making a cavernous angioma form and grow, versus you could have all the genes in the world that cause cavernous angiomas, but if you've got the right microbiome, you won't even form lesions. So some of us have this permissive microbiome, and that's what help allow the lesions to form. But that again doesn't explain why one lesion grows and the other stays small, because your gut should drive all the lesions. So we've been after this problem for some time, and the secret turns out to be in the lesion itself, because everything else in the host or in the patient would drive all the lesions to grow or bleed. So we found out in this research that actually a cancer-like mutation can take place in some of the endothelial cells in a lesion, and then the lesion will catch fire, it'll grow, while the lesions who have everything else the same and the same inherited mutation or whatever will not grow if they don't have that with them. That doesn't mean it's the only factor that can cause lesions to grow, but it seemed we detected it in a large number of cases. So we know that it is a significant player. And then what got the paper into Nature is the fact that the elegant mouse work that Dr. Marshak and Dr. Khan explained to you showed that, in fact, if you get that PIK3CA mutation alone in a vessel, you get some abnormality, but it doesn't go on to a big cavernoma. But if you get a CCM mutation and add to it the PIK3CA mutation, in fact, the cavernous angioma grows huge. And for the first time in that paper, we created a model in mice where it's not the genetic disease, but it's a single lesion disease, like many of you have. You have one lesion with a vein and don't have the inherited mutation. So in fact, when we create through a virus the lesion in the mouse brain in only that spot, without giving the mouse the familial mutation, we can duplicate this. So we can put a CCM gene there and form a small lesion. And then when we add to it in that spot, the PIK3CA activation, that lesion catches fire. And there are some medications that inhibit this activity of PIK3CA. And one of them is rapamycin. And we tested rapamycin in these mice. And in fact, they prevented the lesion from growing and bleeding, as if the mouse did not have the PIK3CA mutation. So you could make up for the PIK3CA mutation with a medication that blocks the effect. So that's what we found. Now, what we did not find are a few things that are important before we can use it in patients. So one of them is the fact that we do not know if your lesion had bled, whether it will stabilize with rapamycin or not. Maybe rapamycin only drives development of lesions like cancer, but has nothing to do with stabilizing the lesion so it doesn't bleed again. So we don't know about that. We also do not know if rapamycin in tolerated human oral doses would have that beneficial effect. So what do I mean by that? So in the mice, to get the proof of concept, we gave them injections in the belly with that drug. None of you is going to get injection in the belly of that drug. Now, we tried to guess what doses would be equivalent to the oral medication, but it's still not a comparison of effect to try to see, is rapamycin even better than just giving vitamin D or propranolol or Tempol or a statin, which all were shown to have effects in mice, and some of them got into Nature papers too. So we can't just go with the excitement of the last discovery as being the drug that will catch fire. This is an important drug. And what we are missing now is doing it on the same mouse models that we had before seen effects with other drugs, and doing what we call comparative effectiveness. We think it will be stronger and more compelling than other drugs, but we don't know that for sure. The other thing is, will it work at low dose? So if you look, many of you have looked up already sirolimus or rapamycin on the internet, and you found that it is approved by the FDA for two indications. One indication is as an immune suppressant for organ rejection. So if you have a transplant, you get given that drug so you don't lose your kidney. And that dose is a very high dose that actually has quite a bit of side effects. It turns out to be better than other immunosuppressants that people with renal transplants have to take. But it's still a very, very powerful drug at those doses. The other indication approved by the FDA is actually for a vascular abnormality. It's both a vascular and muscle abnormality in the lung that is also related to PIK3CA activity. And there, the dose that's approved by the FDA is low, is very low. And in that indication, the side effects of rapamycin are not what they are in the high dose. So the low dose could make an argument that it could be of similar effect to some of the other drugs we have already talked about, and some of them we're trying. Of course, we don't have experience with it in as many patients as take statin, but we may have as much experience as propranolol actually, and we will share with you in due time what that side effect profile looks like in patients who received it for vascular anomalies at low dose. So when you pull papers in the internet, they're not all the same. So the doses are different, the duration is different, but there is a common theme, is that the low dose, which is 1 to 2 milligrams per day in human adult, is actually well tolerated, and we may even be able to do better by giving a drug like this. If it works so well, you could even give it 1 week and have 3 weeks vacation from it. So you take it 1 week a month for 2 years. What does that do? If it really turns off the cavernoma during 1 out of 4 weeks, that may be good enough to quiet it down. and there is already a trial for an inflammatory disease that is using this approach, because for those inflammatory diseases, you also don't want to take a toxic drug. You know, you want to take a drug that's better than the disease. So that's where we are with rapamycin. If I were to guess whether rapamycin will get to clinical trial and when, I would say it's not before 2 years, because it takes probably a year to finalize this data in the mice. I mean, we published what we published because we needed to share the news. And I'll tell you the other important reason that news is important, but it doesn't mean, and we never pretended that we're publishing it to push for therapy tomorrow. The other thing is that in order to get a trial funded, because, you know, rapamycin is now very widely used, and therefore the interest of the drug manufacturer in investing you know, $15 or $20 million to take it to approval is not there like it was a drug they're developing that no one else has. So that's the problem. So it's more of a repurposed drug that is going to be now, you know, some pharma may get interested in it, and that could put it on a fast track, but more likely it is like Atorva and Propranolol. The only way we can get money to test it is through the NIH, and the NIH, you know, takes another year before a trial gets funded. The good news is we have trial readiness with the NIH, and they want a drug to be tested. So the question is, can we sell them that rapamycin is that drug? And today we cannot make that argument because we don't even know it's better than propranolol. We just published a paper that shows how great propranolol works in mice. So, you know, many of you tried it and didn't seem to work as well in humans, but we don't know until we do trials. So that's kind of where we are with that. I want to just say one more thing on the importance of this PIK3CA mutation, is that this now explains why you grow a lesion on a developmental venous anomaly. So all of you who have a sporadic cavernous angioma know that very often you have an associated vein on that cavernoma. Instead of a gene in your body that you inherited from your family, instead you were born with a vein, and then the cavernoma grew on the vein. So what we see there is that the vein is probably the PIK3CA somatic mutation, and then if you get a second hit, then that's where you grow a cavernoma, and the second hit would naturally only take place in the place where you have the first hit. That's why it grows on the vein. And we have some very interesting data on cases that I have operated on where I sampled both the DVA and the cavernoma, and that will be coming out in the literature, and we see where the mutation is to try to prove that. So that may allow us to develop some blood tests that tell which DVA is stable and which DVA is more prone to have other problems. And we are working on those blood tests. And those of you who come to see us, and you know that we collect your blood, this is part of where the discovery is going. So there may be blood tests that will be able to tell us if the DVA is going to stay quiet, or does it have other things that will make it grow. So that's where we are with the rapamycin. I'm sure that generated a lot of questions. So let me move on because I'd like to take all the QAs at the end. So the propranolol story is one that a lot of people have interest in. As you know, people have taken propranolol, and some people said I had great results with it. Other people bled despite it. We don't know how the doses were. We don't know what the indications were for giving it. And if you give it all over the place like this, you may miss an effect because you're not comparing the exact same patients getting it and not getting it. So that's the argument for a trial. in order to get a trial, we had to show that propranolol works in animals, and we didn't have that data until this past year. And there are now 2 studies independently from Europe and from our labs in the US, Chicago and Duke and San Diego, published the study in January that actually shows that propranolol works in mice. The catch is that most people, if they are given propranolol in the dose we gave to the mice, they would be dizzy when they sit up, and they will not tolerate that dose because it may lower their blood pressure. So most people who take propranolol take a much lower dose, about a fourth of the dose we gave to the mice. And now we are funded, thanks to the Be Brave for Life Foundation, to actually do a very quick mouse experiment at the low dose. And that mouse experiment is ongoing. And if we have a low dose effect, then it could make an argument for competing as to whether that is not the drug to test next, if people want a benign drug that's very well tolerated and are afraid of rapamycin. The one who will win will be when we put them head to head. Then we can make the argument as to which is really better and why. And if propranolol does not work at low dose in the mice, there's no way it's going to be funded for a clinical trial in the US. The Italian trial, unfortunately, is going to only show safety. It's not, there is no way, and we agree because I sit on their committee, there is no way that trial was conducted in a way to show an effect. So in order to go from safety to effect, you have to convince someone else to fund it. Maybe in Italy they will fund it, and they will move forward with a phase 2/3 based on their phase 2 data. But that's in the US. We will want an effect in mice on low dose. So where are we with statin? We are ahead of all of these things in statin because we have it in human beings in comparison to placebo. And this last few weeks, we have been very, very busy working on analyzing what we call the interim results of the study. And the interim results have shown so far no futility, meaning that there is a signal effect with this drug that justifies completing the trial. We will redo the futility effect a little bit later this year when 80 people have completed 80 visits. So we are now close to about 60, and a lot of people are logging in additional visits. So we'll get to that, and we'll redo the futility analysis. But it's not going to fail on futility. There is some effect. We don't know if that effect is favorable or that effect is going to be, don't take statin because it's worse. So, and I do not know. We know about bleeds, but we don't know if these bleeds were on statin or on placebo. But an independent safety monitor is looking at this data after every bleed. So somebody knows whether the bleeds are occurring all in the statin and therefore we should stop, or all in the placebo and therefore we should give everybody statin. And this independent monitor has not given us a sign that there is a serious safety one way or the other. So they again want us to continue to collect the data to get the real answer, which needs all the patients. Now, the final thing is, if you took the drug for a year and came for a year 1 visit, but then kind of gave up and didn't come for a year 2 visit, we still will use your data from the year 1 visit. But what happens then is that we have to recruit someone else to make up for that missing visit. So we have built in some missingness, but we didn't build up totally for a two COVID periods where people got shy about their visits and so forth. And there are people who came in late and so forth. So all what I will say is that The data looks very good. I think the missingness is more than made up for by the quality of the data. And there is no need to extend the trial longer. We will, based on the projected numbers, we will meet the power, but there is a small possibility we may be able to complete the trial a little bit earlier based on the quality of the data the way we're analyzing it now. So if that decision is made by the executive committee of the trial, of course, we will make an announcement about it. This will be a public announcement that the trial will complete at 60 or 70 patients instead of the 80 that are planned. And there is an opportunity to do additional analyses with pooling the patients that are in the trial readiness group at Mayo and UCSF and New Mexico, but are not in the trial. But these cannot be used in lieu of the patients in the trial. So these are secondary analyses that may add a cherry on top, but they will not make up. So that we cannot tell you, don't worry, the people have had the test at Mayo Clinic, and therefore you don't need to enroll as many. So the trial itself will only compare people in the trial with placebo versus drug, but we will have additional analyses that are safe to do on the biomarkers, enriching the power of our discoveries. So you bled within a year, your lesion is not resected, you can travel, we pay you for traveling, and we support you through the trial, and many of you are having, it's such a pleasure to log in these year 2 visits and talk you into stopping the medication and all of that. So that is going on, and I think we are doing good there. So it is 6:27. I have 3 more minutes that I want to tell you a little bit about the biomarkers. Very exciting work with the biomarkers. We have evidence now that not only proteins, which we have talked about and published extensively, but there are some other types of molecules in the blood. I cannot quite tell you about them now until they become published, but there are other types of molecules floating around in our blood that will make the test much more sensitive and specific. And the NIH grant for developing the biomarker allows us to use these new molecules. So we're not locked in the old tests that we were doing. So I'm very optimistic on that front, that within the next 2 to 3 years, we have a very robust blood test that will predict who is much more likely to bleed. Of course, people can change their microbiome, or they can go take Botox and bleed, or do something and overdo the biomarker. But in general, that biomarker is going to be much more predictive of stable cases and unstable cases. And it will also help us answer the question many people have. I had a headache 6 months ago. I didn't have an MRI. Did I bleed? Now that question is important, because if you really bled and we didn't pick it, that lesion is more likely to rebleed. So if you get a test that can help answer some of these questions, even when the MRI is late or done not well, or not done at all, then that again will help us separate people who bled from people who didn't bleed, and that's an important differentiator. It's not the only differentiator. People have headaches and seizures without new bleeds, but we estimate that new bleeds account for about 80 to 85% of the cumulative disability of this disease. So that's kind of where we are, and I finished 1 minute ahead. Have you ever seen me finish 1 minute ahead? So I think we're good.
I'm very impressed. Very impressed.
All right. So
okay.
Let's take questions.
I have actually not a question from the audience, but one more topic, which is COVID vaccines. I continue to get questions about COVID vaccines and safety for patients with cavernous angioma. Do you have any indication that they might not be safe, or any other words?
Yeah, so as you know, we have a very large registry, the largest in the country, of patients in our practice. The CCM registry that we did together, Connie, with the CCM community is only designed to look at severity of illness with COVID. So if you have a COVID infection, and did you bleed, did you not bleed, did you go to the hospital, did you not go to the hospital, that there are going to be some answers coming up from that. And the summary of that is we don't recommend that you get COVID infection if you have a CCM. Okay, it's not good for CCM. It's not just not good because you can die from your lungs, but you may actually be bleeding at higher rates if you get a COVID with CCM. So, hear me out on this. Anything we can do to prevent COVID is good. So we have not seen any signal effect of brain bleeds or CCM bleeds related to the vaccine. Have not seen it, and I look for it because we ask the question, are you vaccinated, to everybody, just so I can make my preaching thing. And so we collect that data on everyone who sees me, and we did detect an effect of increased bleeding with illness, but we did not see any increased bleeding in the 50-fold more cases who got vaccines. So there are a lot more people who got vaccines than bled. And if there was an effect, I'm confident we would have seen it. So please get vaccinated. That's the correct scientific answer. You may have other reasons. I'm not going to go into politics of vaccines, but the science is very clear that you should get vaccinated to protect yourself and your family and the community in whom you live.
Thank you. All right, now on to the questions. I'm going to start with the PIK3CA questions and try to group those all together. Here's a very good question. Is there a way to test to see if patients carry the PIK3CA mutation?
Yeah, so there are two ways to test for it. The best way you don't want, because that's in the lesion itself. So basically you biopsy the lesion and look for it, because these are somatic mutations. They're not mutations you inherit. So you cannot go to prevention genetics and test for PIK3CA. So if you inherit PIK3CA, it's really bad. You're going to get cancers long before you get Cavernous. So we're not talking about people inheriting that mutation. We're talking about people who get it in that lesion. So no, you cannot find out if your lesion has it or not unless our new biomarkers detect that signal in the blood. And we, again, I cannot tell you about it now because it's not published yet, but there are signals of molecules that we have identified that are new, that have not been talked about before, that only occur in the people who have PIK3CA. So that could be a very good test for that somatic mutation in the blood. But we're not quite there, but we know why you're asking and we're working on it.
Okay. All right, next question, also good. So can you please tell us, if this is a comparative question, the mechanism of how rapamycin works versus propranolol versus statins? How are the mechanisms different?
Yeah, very good question. So propranolol works as best as we know on the receptors in the red cells that respond to pressure and flow. So it basically works by the similar mechanism of lowering flow, lowering blood pressure, kind of chilling out the flow in the lesion. And that's what happens in the fish, but then in the mice, the same thing is happening. Whether that's the case in humans at low dose that don't lower your blood pressure so you're not dizzy. The mice can't tell me if they're dizzy or not, you know, so it works more on blood pressure and flow related mechanisms. Statin is a very interesting drug because it works downstream of all of these. So basically, it will inhibit the effect of PIK3CA activity. It will inhibit effects of KLF4 because all of those are upstream and they activate ROCK. so the vessel doesn't leak and doesn't bleed unless ROCK is activated. So we're working with statin on the downstream, not only statin, but all the ROCK inhibitors, bioaxons, drug, etc., are working on this premise that it doesn't matter what signal drives it upstream, you stabilize the lesion by stabilizing the blood vessels. So that's how they are different. Now, rapamycin works on inhibiting the signal of that PIK3CA activity. So it's like taking the turbo out of your Porsche, you know, then it becomes a regular Porsche, but it's not turbocharged anymore. So that's how rapamycin works.
I'm giggling because Doug Marchuk and Mark Kahn both used car analogies too. You guys
Here you
talk to
go.
each other.
But I know they don't have the Porsche I have.
No, they don't. No, they were like gas and handbrake. I mean, they were much more basic. All right. Then more of a business side question. Is there any possibility that either rapamycin or propranolol could be reformulated to optimize and thus generate a new molecule that could attract investment in a trial? Do we know the chemistry of the drug to investigate that possibility?
Yeah, so there are two arguments for repurposing other than the obvious business question, because if you create a new molecule, then you have a patent on this molecule that you are the only one who benefits from it for at least 10 years and can be extended more if it's a rare disease. So there is a lot of interest in pharma and new molecules. But they cannot just say, I want to do a propranolol and kind of add a little feather on it, and then they'll give me. So the actual patent has to be that you made it better than propranolol. Okay, and that's a big tall order, you know, because we just discovered that propranolol works. So how the hell are we gonna, you know, and who's going to invest in changing the molecules and seeing how much more it helps or not help, unless those people are developing the drug. So there is a group in pharma that are doing the computer work on this. And there is a big industry of this in India, and they are very, very good at this. So they take a molecule and they look at it chemically and try to say, well, if I changed it a little bit, maybe now I have a new drug. And so I'm sure there are some pharma that are looking a little bit at this. Every time we publish a paper, there are a few businessmen that look at it from that angle, but it's just not there now. Now, an example of that is the ROCK2 inhibitor story, because that developed with the idea of getting a drug that's better than statin, right? So the whole idea was we want to get a drug that works more in the brain rather than in the systemic effects, and see how it might or might not help. The only problem is that even in the mice, it didn't do better than statin. It just replicated statin. So you know, pharma is going to have to wait to see if statin works. There is no way they're going to spend $60 to $80 million developing a new drug while you could buy one for 10 cents and do the same thing. Mm-hmm. So the whole issue is, are you really developing a better drug?
Okay. Next question. We know that radiation can cause the development of lesions, of cavernous malformation lesions, even in people who are just getting the radiation for brain cancer. How do we have any understanding of how the PIK3CA mutation, is there any relevance there? Is there
so PIK3CA is not the only oncogene that drives cavernomas. You're gonna read papers in the next year that have other genes that do similar to PIK3CA. And therefore, what radiation is doing is making that spot where the radiation goes have more mutations, and therefore more likely that one of those bad mutations would happen and the lesion would take off. So there is no question that radiation increases the rate of second hits. So if you are going to get a PIK3CA somatic mutation by chance, you are definitely more likely to get it when you radiate.
All right. I think that is all I have for specific to rapamycin PIK3CA. So a biomarker question. Will the biomarker blood test be relevant to both familial and sporadic forms?
Yes. So far, the results we found shows equal effectiveness in both groups, but the groups were small, and we didn't have individual genotypes to be able to tell you, is it as good in CCM3 as it is in CCM2? Because they have relatively different lesion burdens, and therefore the biomarker power may be different. The good news is that the current NIH trial, it's already funded. So we don't have to go ask for money or anything. It's already funded to tune of about $4 million, is funded to collect enough patients to answer that question. So we're working with Mayo Clinic, with UCSF, and maybe with one additional center, plus also off the grant with the center in Brazil, to try to test it in these different environments with different types of patients. And one of the fundamental questions we will control for is familial versus sporadic. And it may end up being that the test is tweaked a little bit. The formula is tweaked a little bit to make it better for the familial than it is for the sporadic. Could be tweaked a little bit for women or for older patients. So we're looking after that. When I talk about tweaking the tests, the companies that want to produce tests, they quench. They don't want that. They want a canned test. So they want us to develop the best test that works across the board, so they know what they can go and put in the can to sell. And they don't want that every day, I talk to Justine and develop a test for her. I talk to Connie, develop a test for her. I talk to Connie's daughter, develop another one. So the issue here is going to be that we will want to develop not just the best biomarker for individual patients, but also the best biomarker that can be developed across the board. And there will be some molecules that are in common across genotypes and ages and sexes.
All right, and along those lines, someone else asked, would the biomarkers be just for adults, or could children under 6 or 7 use these instead of MRI with sedation?
Yeah, so very, very good question. And there is a little bit of a misunderstanding as to how biomarkers would be used. I think a biomarker will never tell you for sure if your kid has a lesion or doesn't have a lesion, or if they get double vision, whether they bled in that lesion or not. We have MRI for that. So it is not a compelling argument to develop it to replace MRI. However, it will enhance and maybe cut down on MRIs, which is extremely important in children. There are some parents who elect, as you know, not to do the first MRI on their kid unless they have symptoms. Mm-hmm. So it will be very powerful to have a biomarker that shows that. Unfortunately, the original development is for 18 and older, because that's how we're going to get it to the pharmas and to the FDA. But nobody is missing the CCM3 question and the kids, because we want to apply it in children. This is on our radar.
Okay. All right. On COVID, will there be data published on the cases of CCM bleed and COVID?
Yes.
Okay. They're working on it. This sort of more toward microbiology.
But I can't show it to you tonight, because then nobody will publish it.
I know.
I'm just telling you enough of what I told you. Don't get COVID, get a vaccine.
Right.
Okay, next.
Has there been any investigation into whether cavernomas have anything to do with diverticulosis?
No, there hasn't been. Now, there has been a lot, as you know, related to inflammatory bowel disease. We've had a hypothesis related to that. It is compelling that inflammatory bowel disease will cause leak in the gut. Diverticulosis is a different disease. It is not a permeability disease. It's more of a growth on the side of the gut, and that gets inflamed. We don't know if that changes the microbiome enough to affect CCM. So there is no data on this.
Okay. All right. And then I have a few people asking about Botox.
Yeah.
One person had a small brainstem lesion that bled 3 years ago and wants to know whether it's safe to use Botox for cosmetic reasons, just a little bit. And another person says they are using Botox currently for migraines. And is that a safe thing for someone with cavernous malformation to do?
Yeah. So the evidence on Botox is what we call anecdotal. So basically, you come and you see me in the office and you had a bleed, and we talk about what has happened prior to the bleed. And some things occur by chance, and some things occur only in the people who bled.
Mm-hmm.
So in vitro fertilization and bleeds is a big commonality. A huge exercise, they're going, I used to joke always climbing Kilimanjaro and then something happens. Or a new yoga that I've never done before. So the Botox evidence fits with that. So we have looked at Botox, and it accounts for a number of bleeds where there is no other explanation, and we don't see it in the people we don't see that same signal in the people that don't bleed. So I don't have many people who haven't bled who get Botox all the time. But about once a week or once every other week, the only factor that comes up in the clinic is that I just got Botox. And nothing else explains my hemorrhage. Now, does it make sense biologically? Because you don't want to just say, my husband smoked a cigar and I bled, So it's not like that, because Botox is linked to the TL4 signaling, which is the very signaling that drives cavernous angiomas.
Mm-hmm.
So you can't make that up. It's a toxin that activates TLR4. So yes, I don't have a study that has 100 patients who got it and didn't get it. And what I tell people, it's the same risk benefit that you've got to take when you make any decision in life. I can only tell you that it has caused bleeds, and you have to decide if little is little enough. Are you going to tolerate a little bleed or a low risk of a bleed or whatever? But there is nobody that's going to tell you that it is 100% safe in cavernous angiomas after we showed the data. It's just not, nobody will tell you it's 100% safe. We will only do a yellow light for you to be very careful, very judicious. And certainly if you bled after a Botox, cut out Botox. Please don't do it again, because the case we published, every time she got the Botox, she got the bleed after that. It was like the story of her life.
Okay. Next question harkens back to something you and I have discussed recently. I was diagnosed with CCM and have more than 20 cavernomas. After testing the 3 mutations, CRIT1, CCM2, and PDCD10, my tests came up negative. They said I might have another mutation that hasn't showed. And then separately, the doctor put me on Atorvastatin for the last 2 years. Can I get a second opinion? Are there any other mutations that you know of?
Yeah, so let me first answer the Atorvastatin. There is no reason to be put on Atorvastatin for CCM or CCM-like disease today. There is only evidence to go into a trial to find out whether it works, because it could make it worse. So nobody can tell you it's safer than the placebo until we complete the trial. So we see a signal, but I don't know what to tell you on that until we break the blind. So that's that. Now, you may have been put on statin for cardiovascular indications related to cholesterol or preventing a stroke or something like that. Those indications are okay. And in fact, we don't want you to stop the statin either. If you've got a stent in your heart and you have a statin or carotid lesion or high cholesterol and you're on statin for that, I never stop it. So we don't stop statin for that. So if you're on it for those reasons, that's fine. If you're on it for your bleeds in the brain, we don't know if you're making it better or worse. So the next thing comes up as to whether your disease is even CCM. So many people can have an entity called hemorrhagic microangiopathy, which has small bleeds, big and small. They can be caused by high blood pressure, etc. And other factors like congenital heart disease can cause that. People who had that bypass surgery can get it. And after radiation, like Connie said. So after radiation, there are some cases that are true cavernomas and some cases that are just bleeds. When we resect it, there's no cavernoma, there's just blood there. And therefore, differentiating microbleeds from cavernous angioma is very hard to tell. The only way I can tell you you have that fourth gene we don't know about is if also you have a family history of multiple cavernous angiomas, proven, not by rumor. So if your sister or dad, a first-degree blood relative has multiple lesions, if those multiple lesions actually look like cavernous angiomas more than microbleeds, and the good doctor can tell the difference. Recently, Connie sent me some pictures, and I looked at that as an example. I couldn't give medical opinion to patients, but that's so a good doctor can tell a differential diagnosis and tell you. But I don't think it's a foregone conclusion that you have familial with another gene, and there is no way you can test for it since there is no mutation that you found.
Right.
So,
And
what are you going to test for?
Right. And you had also mentioned to me that there was a 5% false negative.
Yeah. So false negativity is all those causes combined. Mm-hmm. So false negativity, the lesions are not multiple. They just were told they're multiple, and actually all three are near a vein. We talked about that. So a person who doesn't know CCM as well as Dr. Fleming or myself will call it multiple. Okay, and then you're carrying that and you're thinking I have the familial. So multifocal is more correct than multiple and without a vein next to them. So that's one thing. Is it truly familial? Or are you worried about your family, but it's not familial? So if you're worried about your family, the only way is that if there is a gene you test for. Otherwise, you have to say, you know, how much do I want to do MRIs on every baby who's related to me, you know? And our recommendations in the guidelines is that if you are mutation negative, that you only do MRIs for neurologic symptoms. It is not fruitful to do MRIs on kids related to somebody who doesn't have a mutation if they test it.
Okay. All right. 2 questions related about sleep. One person has had trouble sleeping since surgery and wondered if there was any advice. And then the other person, follow on, is, are sleeping medications like Ambien shown to be an issue for CCM?
Yeah, so, you know, sleep is going to become interesting. That will be the next wave of webinars we'll do because of the issue about hypoxia and sleep apnea and so forth. So let me tell you what we know so far. So we know that in the mice, if you give them low oxygen, even intermittently, like somebody might do when they're snoring or having sleep apnea if they're obese, those mice will have more lesions. However, when we looked at the human data in the familial CCM database, both in the BVMC as well as in our own experience, it doesn't correlate with the number of bleeds and lesions you have in your head. And there is a very simple reason for that, is that the lesions may have formed and your bleeds may have happened before you got sleep apnea. Most people get sleep apnea later in life. They're not getting sleep apnea when they're 20. but they may have gotten their bleeds and their lesions to form before there was sleep apnea. So we have to do prospective studies on the people with sleep apnea and without to see if they are more likely to bleed or form new lesions. But it's not as easy as just checking 20 people and saying you have sleep apnea as your disease worse. So that's that. Now, I know that wasn't your question, but the question about hypoxia will relate to this issue. So you don't take oxygen to try to treat the lesion. I just would avoid hypoxemia as much as possible if we can.
Okay.
That answer the question?
No, but that's okay.
Go ahead. You want to
There were two questions. One of them was, someone is having trouble sleeping since surgery, and I know this person.
Yeah, that is.
I think that it was.
I don't know where the surgery is.
Well,
I don't know.
Frequently we have patients with brainstem surgeries who have sleep problems after surgery because of sleep centers being located in the brainstem. And the question was, what do you typically recommend for patients who are in that situation?
I think you need to study the sleep problem, go to a sleep specialist, and they will do some analyses on you as you're trying to fall asleep and why you're restless at night and so forth, and try to figure out, is this some reflex that's gone bad or whatever. I must admit, I haven't seen that particularly in all my experience with brainstem surgery. I've seen it with a lot of surgeries that people have that. So it may be, sleep is such a broad symptoms that unless you go to a sleep specialist that diagnoses your sleep problem, there is not going to be a single answer across the board.
Yes, agreed. All right. Should someone with a cavernoma avoid having electroconvulsive therapy, ECT?
So what does ECT do? So ECT basically puts a big jolt in the brain. And if anybody has seen a patient getting ECT, which I have, and of course those who have had it know what they go through, and my hat is off to them. It's a very traumatic experience where there is a lot of shaking that happens, and we just do not know the effect of that stress on the blood flow in cavernous angiomas, whether it makes them more likely to bleed or not. We just don't have enough data. My first initial impression would be, I would avoid that type of trauma if you've had a lesion that recently bled, for sure. If all your lesions are stable and there is nothing else working for your depression, you might want to take that risk. But certainly someone who bled recently, I'd be hesitant.
Okay. I have questions that are coming back to some of our past topics, and we're pretty much at the end here. We're 2 minutes out, so rather than trying to go back and revisit some things, why don't we just close out for the evening? Is that okay?
Yeah,
with you?
yeah, it's perfectly fine, and I think it's right on time. So I want to really thank everybody, and I want to just tell you that you can email cash@uchospitals.edu for any research question related to cavernous angiomas. So cash@uchospitals.edu, and or go to our website and you click on the research and you have contact with our coordinators. So we're here to try to answer you. We answer a lot of questions, even on people who we are not seeing clinically, but of course we cannot give you medical advice.
All right. Thank you so much for your time. It was good to meet Justine and see Agnieszka again, and I want to wish you a wonderful vacation. I know you're going to be away for a while now.
Thank you very much, everybody. But we're available to you, and even when I'm on vacation, the team is busy like you wouldn't believe. Okay, thank you, guys.
Bye-bye. Good night.